You walk into a modern hospital, right? You smell the bleach, you rub that alcohol hand sanitizer into your palms, and you just assume you are in this completely sterile, engineered safe zone.
Yeah, that's the illusion, definitely.
Right, but if you actually zoom in to a microbial level, that hand sanitizer you just used did absolutely nothing, like zero to the dormant spores of Clostridioides difficile that are probably covering the surfaces around you.
Which is a terrifying thought.
It really is. You haven't entered a safe zone at all. You've basically stepped into an evolutionary war zone.
Mm-hmm.
And today we are doing a deep dive into a really comprehensive, peer-reviewed clinical microbiology review paper that details the history, epidemiology, and the cutting-edge treatments of C. diff.
And the scope of this source material is just incredible. I mean, it gives us this molecular level view of how the bacterium actually operates. It meticulously tracks how a pathogen that we barely even understood a century ago managed to just outsmart our most advanced medical interventions.
And it did it by exploiting the very treatments we use to heal people, which is just wild.
It-
So, our mission today is to extract the most mind-blowing and crucial insights from this research. We want to give you a shortcut to understanding how a once rare surgical complication evolved into this global healthcare terror, and how the future of medicine is actually using the human microbiome to fight back.
It's a massive shift in how we think about treatment.
Okay, let's unpack this. Because the timeline of C. diff reveals a really massive blind spot in early medicine. Like the first description of the disease, it causes pseudomembranous colitis that was documented way back in 1893 in a 22-year-old woman after gastric surgery.
Right.
Yet, when C. diff itself was finally isolated in 1935, researchers just completely dismissed it.
Yeah, they basically considered it a normal, harmless part of the neonatal intestinal flora, just, a background
That's
microbe.
all worry about.
Exactly. It really wasn't until the 1970s that the scientific community realized what they were actually dealing with, that it required a huge paradigm shift. Clinicians were seeing severe colitis in patients who were taking the antibiotic clindamycin.
Right, the antibiotics were somehow triggering it.
Yeah, and the breakthrough came via what's called the hamster model. Researchers gave clindamycin to hamsters, which predictably wiped out their normal gut flora.
Makes sense.
But in that ecological void, a specific toxin-producing Clostridial strain just rapidly multiplied, and that's what caused the fatal colitis.
So the antibiotic wasn't directly damaging the gut itself, it was just like clearing the competition.
Precisely. And they proved this by introducing a different antibiotic, vancomycin, which selectively targeted the Clostridial strain and actually protected the hamsters.
Oh, wow. So the paper marks this as the exact moment we finally linked the pathogen, the mechanism of dysbiosis, and the treatment.
Yes. But, of course, the pathogen immediately started adapting to our countermeasures.
Naturally.
The review highlights this really critical evolutionary leap in 2003 during an outbreak in Quebec, Canada. The incidence rate just surged from 35 to 156 cases per 100,000 people.
That is a huge jump.
It was. And even more alarmingly, the mortality rate nearly tripled, going from 5% to 14%. We were basically witnessing the emergence of the hypervirulent NAP1027 strain.
The superbug.
Exactly. This variant had acquired a mutation that gave it extreme resistance to fluoroquinolones, which is a really broadly prescribed class of antibiotics.
Okay, so the best way I can think to visualize this dynamic within your own gut microbiome is to picture a dense ancient forest.
I like that analogy.
Right. You have thousands of competing microbial species, and they're all kind of keeping each other in check. But when you take broad-spectrum antibiotics, like those fluoroquinolones, it's like dropping a match in a dry summer.
Total devastation.
Exactly. A wildfire just sweeps through, incinerating the canopy, the underbrush, everything. And C. diff is basically this deeply buried fire-resistant weed.
That is a great way to put it.
Yeah, so with all the biological competitors gone and the ecological resources just completely uncontested, this weed rapidly colonizes the barren landscape.
If we connect this to the bigger picture, the Quebec outbreak really demonstrates that C. diff is primarily a nosocomial or hospital-acquired infection that is driven by our own medical protocols.
So we did it to ourselves.
We really did. We inadvertently engineered an evolutionary arms race. By saturating hospital environments and patients with broad-spectrum antibiotics, human medicine applied the exact selective pressure needed to cultivate a hypervirulent, highly resistant superbug.
It's just crazy to think about. And the geographical data in the paper maps out exactly where this superbug is thriving today. And it is far from an equal opportunity pathogen.
Not at all.
North America registers significantly higher rates of C. diff than Europe, and the divergence really comes down to the microscopic terrain we're creating inside ourselves. Like, if you're looking at your own daily habits, the paper suggests your diet is fundamentally altering your localized risk.
The diet connection is huge.
Yeah. It points out that the Mediterranean diet, which relies heavily on plant-based carbohydrates and is lower in animal protein, acts as this profound defensive barrier.
And the mechanism behind that defense is entirely microbial. A Mediterranean diet actively lowers the ratio of firmicutes to bacteroideates in the human gut.
Okay, so those are two major groups of gut bacteria.
Right. And maintaining that specific microbial balance significantly increases the production of short-chain fatty acids, primarily one called butyrate.
Butyrate?
Yes. Butyrate is actually the primary energy source for your colonocytes, which are the cells lining your colon.
Oh, interesting.
So by feeding those cells, the short-chain fatty acids fortify the gut epithelial barrier and create a localized chemical environment that indirectly inhibits C. diff proliferation.
So a strong wall keeps it out.
Exactly. Conversely, the high-protein, high-fat Western diet basically starves those beneficial microbes, reduces the butyrate production, and weakens the colon's structural integrity.
And the environment isn't just weakened, it's actively seeded with the pathogen. The contamination statistics comparing global food supplies in this review are just staggering.
They really are.
Like in Europe, roughly 3% of retail meat samples test positive for C. diff contamination. But in North America, up to 42% of meat samples harbor the spores, specifically those hypervirulent 078 and 077 strains.
That's nearly half the meat tested.
Right. And for those assuming a pescatarian diet gets you out of the woods, it doesn't. The global seafood supply carries a 10.3% contamination rate.
Yeah, you really can't escape it.
Combine that food supply with the fact that the U.S., France, and Italy remain among the highest global consumers of broad-spectrum antibiotics, and you literally have a population actively priming its gut ecology for an infection.
And the overlapping ribotypes between the agricultural sector and human patients validate this shared ecological reservoir. Researchers isolate the exact same genetic 078 strain in commercial piglets, on retail meat packaging, and in the intensive care units of nearby hospitals.
Wait, hold on. I have to challenge the direct transmission angle here.
Okay, go for it.
If 42% of our meat has C. diff sitting on it, and the spores survive cooking temperatures, are we literally routinely getting infected directly from our dinner plates? Or is the contaminated food simply a downstream symptom of a larger variable, like agricultural sectors using massive quantities of antibiotics that select for these strains in the livestock?
That is a fantastic point, and the source material positions that exact question right at the frontier of current epidemiology.
Okay, so what's the consensus?
The consensus actually leans toward the latter. The widespread agricultural use of antimicrobials creates this immense evolutionary pressure that amplifies these specific ribotypes across the entire food chain.
Ah, I see.
So yes, you are likely ingesting spores regularly, but ingestion alone doesn't equal infection. The pathogen still has to navigate the human digestive tract, which requires a highly specialized survival mechanism.
Right, because C. diff in the external environment doesn't operate like a standard vegetative bacterium. It circulates as a metabolically dormant spore.
Like a tiny armored tank.
Exactly. The core of this spore is heavily dehydrated and encased in this complex of calcium and dipiclinic acid, or calcium DPA. And this architectural armor renders it basically impervious to ultraviolet radiation, extreme heat, and most chemical disinfectants.
Which loops right back to the hospital hand sanitizer issue we mentioned at the start.
Yes, alcohol cannot penetrate that calcium DPA layer. The standard protocol requires vigorous mechanical friction with soap and water to physically dislodge the spores from your epidermis and literally just wash them down the drain.
The armor is brilliant for environmental transit, definitely, but the pathogen cannot produce toxins or replicate while it's dormant like that.
It has to wake up.
Exactly. It must germinate, shedding the calcium DPA casing to become a metabolically active cell, and it does not shed that casing randomly. The germination process is precision engineered to only activate when the spore has successfully reached the nutrient-dense environment of the lower intestine.
Okay, here's where it gets really interesting. The spore passes through the extreme hydrochloric acid of the human stomach without registering any damage at all.
None.
It stays entirely dormant until it detects specific biochemical passwords. And those passwords are your own bile salts.
It's almost malicious, isn't it?
It is. The source specifically identifies cholic acid derivatives as the primary triggers. So the spore relies on the host's own digestive fluids to signal that it has safely arrived in the colon. Your body is biologically signaling the assassin to unpack its weapons.
It is an exquisite evolutionary hack, but the review uncovers an even more concerning catalyst regarding common everyday medications.
Oh, right, the antacids.
Yes. We know antibiotics clear the biological competitors, but the data points to proton pump inhibitors, or PPIs like omeprazole, as a massive independent risk factor.
And the clinical assumption has long been that PPIs increase risk simply by neutralizing stomach acid, right? Like allowing more bacteria to survive the initial ingestion.
Right, but the calcium DPA spore already survives the stomach acid anyway.
Exactly, so the acid neutralization is completely irrelevant to the spore.
What's fascinating here is the insidious downstream mechanism of the drug. Omeprazole induces profound gut dysbiosis, fundamentally altering the microbial architecture of the lower intestine.
Wow.
Furthermore, the paper details how the medication directly interacts with the bacterium's metabolic pathways. Omeprazole alters purine metabolism within C. diff.
So it's feeding it.
Basically, this metabolic disruption acts as a stressor that aggressively promotes sporulation. It also upregulates the bacteria's flagella, significantly increasing cell motility.
So you have a ubiquitous over-the-counter medication fundamentally rolling out the red carpet, accelerating the pathogen's ability to move, multiply, and synthesize toxin A and toxin B.
It's the perfect storm.
And once those toxins are synthesized, the physical destruction of the colon begins. They trigger the severe inflammation and watery diarrhea associated with the disease.
It's brutal.
But the clinical data shows that patients with hypoalbuminemia, which is low levels of the protein albumin in their blood, are at a drastically higher risk for severe fatal complications.
Which is really surprising on the surface.
Yeah, because usually we just think of albumin as a standard transport protein maintaining osmotic pressure in our veins. But the paper reveals it plays a direct physical role in the immune response here. It acts less like a passive transporter and more like a molecular decoy.
A decoy.
Yeah, human serum albumin binds directly to toxins A and B. Because the albumin physically occupies the binding sites on the toxins cannot latch onto the host's epithelial cells to trigger cell death.
And the experimental data validating that decoy mechanism is remarkable. Researchers utilized zebrafish embryos, exposing them directly to C. diff toxin B.
Okay, what happened?
The baseline mortality rate was 50%. But when they introduced human serum albumin into the environment, the albumin bound the free-floating toxins, dropping the mortality rate to 30%.
It's a huge drop.
It is. It provides a clear mechanical explanation for why maintaining high serum albumin is a critical defensive parameter for infected patients.
But wait, that zebrafish mortality metric forces us to completely reevaluate the scale of the disease? Like, if toxin B is lowering the heart rate and killing zebrafish, we aren't just looking at localized colon damage anymore.
No, we're definitely not.
Once toxins A and B degrade the epithelial barrier in the gut, the colon becomes highly permeable. That leakiness allows the toxins to breach the bloodstream, shifting this from a localized gastrointestinal issue to a massive systemic crisis.
And the detection capabilities have finally caught up to the pathology to prove it. Using highly sensitive novel assays, clinicians discovered that 94% of patients with the infection had detectable toxemia.
94%.
Yes. The toxins are circulating systemically in almost every symptomatic patient. Animal models demonstrate this circulating toxemia directly induces cardiovascular damage, triggers widespread renal inflammation, causes atrophy in the thymus, and even breaches the blood-brain barrier.
The blood-brain barrier, that is terrifying.
It really is. The systemic reality dictates that we can no longer rely on brute force antibiotics that cause further collateral damage.
Right, and the pharmaceutical strategy is evolving to reflect that. Like we mentioned, for decades, the gold standard was vancomycin.
Which works, but
vancomycin still causes moderate to high disruption of the surrounding bacteridases and firmicutes. So the cutting edge of treatment is the shift to a drug called fidaxomicin.
Fidaxomicin is fascinating.
Yeah, the paper explains the mechanism that makes it so valuable. It acts like a sniper rifle. It specifically targets the RNA polymerase unique to Clostridial species.
Ah, so it binds to that specific enzyme.
Right. It shuts down the pathogen's ability to synthesize proteins. And because it targets a Clostridial-specific pathway, it largely spares the surrounding healthy microbiome. It basically kills the target without burning down the surrounding forest.
And the preservation of the native flora is why fidaxomicin yields significantly lower recurrence rates than vancomycin.
That makes total sense.
Additionally, by maintaining the ecological pressure in the gut, Fidaxomicin actively reduces the patient's susceptibility to acquiring other opportunistic nosocomial pathogens, such as vancomycin-resistant enterococci.
So it handles the immediate biological threat while preserving the environmental defenses against the next one.
Exactly. But even with RNA polymerase targeting, a subset of patients experience endless recurrence loops.
Right, their endogenous microbiome is just too thoroughly decimated to repopulate and hold off the remaining spores.
Yes. And the ultimate clinical escalation there is fecal microbiota transplantation, or FMT, transferring donor stool into the patient's colon to restore the microbial balance. It's actually been utilized since 1958.
Right, and the logic has always been really straightforward.
Yeah.
You introduce live, healthy bacteria to outcompete the C. diff for space and nutrients.
And this is a massive, but the review cites a study by Ott et al. That completely dismantles that logic.
Wait, hold on.
They utilized a sterile fecal filtrate. They took donor feces and filtered it so aggressively that absolutely zero live bacteria remain. Zero. They transferred a sterile liquid into the patients, and it still successfully cured the C. diff infection.
Wait, hold on. If you filter out all the actual good bacteria and just use a sterile liquid and it still cures the patient, then the whole replanting the forest analogy I used earlier is fundamentally wrong. What biological mechanism is actually executing the cure?
This raises an important question, one that really forces a paradigm shift in how we define a microbiome. The source material hypothesizes two primary mechanisms operating within that sterile filtrate.
Okay, what's the first one?
The first is bacteriophages. These are specialized viruses that exclusively hunt and infect bacteria. The donor filtrate is saturated with phages that seek out the C. diff cells, inject their viral genetic material, and cause the bacterial cells to rapidly lyse or burst open.
Wow, so we aren't just transferring a competing ecosystem, we are transferring viral predators.
Precisely.
That is incredible. And the second mechanism relies entirely on the chemical footprint left behind by the donor's bacteria, right?
Exactly. The filtrate retains the dense concentration of metabolites produced by a healthy gut, specifically the short-chain fatty acids and antimicrobial peptides we discussed earlier.
Like the butyrate.
Yes. Even absent the live bacteria, this concentrated chemical payload is sufficient to rapidly restore the host's epithelial barrier. Furthermore, these metabolites immediately alter the local chemical gradient, shifting the bile salt composition in the colon.
Oh, they scramble the biochemical passwords.
They do. By changing the bile salt profile, the environment no longer signals the C. diff spores to germinate. It effectively locks them in their dormant state until they are flushed from the system.
So the chemistry of the microbiome is proving to be as potent as the microbes themselves.
Without a doubt.
So what does this all mean? The trajectory of Clostridioides difficile is basically a masterclass in unintended consequences.
It really is a cautionary tale.
Yeah, it reveals how a healthcare system heavily reliant on broad-spectrum antibiotics to neutralize infections and powerful acid suppressants to manage indigestion inadvertently engineered the ultimate biological niche for a superbug.
We created the monster.
We did. We systematically cleared the ecological competition, provided the precise biochemical triggers required for germination, and facilitated its spread across vulnerable populations. Yet the research proves that the solution isn't found in deploying increasingly destructive chemical weapons.
No, the brute force approach is over.
The future of treating complex pathogens requires a deep integration with our own biological ecology, utilizing highly selective mechanisms like fidaxomicin, or leveraging the viral and chemical architecture of a healthy microbiome to outmaneuver the disease.
The clinical framework is transitioning from sheer eradication to sophisticated ecological management. It demands we view the human body not as a sterile vessel, but as a dynamic, deeply interconnected habitat. If our own digestive fluids act as the catalyst for pathogen germination, and our daily dietary choices dictate the structural integrity of our immune barriers, we must recognize that modern humans are actively terraforming our internal environments. We assumed the evolutionary war was happening on the hospital surfaces around us, but the primary battleground has always been our own altered ecology.
Wow.
And if our current habits perfectly prime the environment for this specific superbug, what other dormant threats are quietly waiting for the next dietary shift or the next wave of antimicrobials to finally wake up?